Hi, I'm Dr. Pardo at Imperial College London, and today my colleagues and myself are going to tell you about our latest story. My team is based at the Division of Cancer on the Hammersmith campus of Imperial College, and our work has mostly been focused on lung cancer, which is the principal cancer killer worldwide. In particular, we want to understand what makes this disease become resistant to therapy and prone to disseminate in the body. Indeed, the development of drug resistance and metastasis are the two main reasons for failing to cure lung cancer patients. So for several years, my team has worked on elucidating the molecular mechanisms responsible and most recently started work on an enzyme called RSK4. The first thing that we noticed was that RSK4 is overexpressed in lung cancer samples as compared to normal lung, and lung adenocarcinoma patients with higher expression of this enzyme had a significant decrease in survival. So we wondered whether targeting RSK4 could be beneficial to patients. We first looked in animal models where lung adenocarcinoma cells grow efficiently into tumors and respond poorly to cisplatin, one of the drugs used to treat this cancer in the clinic. However, ablating RSK4 expression prevented efficient growth of the tumors and moreover sensitized these to drug treatment. Also, while we found that cancer cells colonize the lung of animals quite efficiently, ablating RSK4 almost totally prevented this. So we investigated what other proteins RSK4 interacted with in the cell and elucidated how RSK4 mediated its effect on drug resistance and metastasis. We found that RSK4 impacted on two separate pathways. First, RSK4 promoted the expression of a pro-survival protein called CIAP2, and this resulted in the inhibition of cell death and subsequent response to drugs. Separately, through a series of modifications on constituent molecules, RSK4 increased the transcriptional program downstream of NF-kappa B, which resulted in cellular changes to promote cell migration and invasion.
So you've just heard from my colleague Dr. Pardo that RSK4 plays a critical role in regulating metastasis and sensitivity to chemotherapy in lung cancer cells. We found exactly the same thing in bladder cancer cells, and this prompted us to then consider that targeting RSK4 could provide a novel therapeutic approach for the management of patients with these cancers. But the first thing we needed to do was to find a small molecule inhibitor for RSK4. Therefore, we performed a drug screen and identified some members of the floxacin antibiotic family, and in particular trovafloxacin, as potent RSK4 inhibitors. Treatment of lung cancer animal models with trovafloxacin reproduced the effects seen following genetic ablation of RSK4 and resulted in improved response when combined with chemotherapy. We then worked with colleagues in the Mathematics Department and Department of Life Sciences at Imperial College to determine the mechanism through which trovafloxacin inhibited RSK4. This work will now enable us to further improve the RSK4 compounds to make it even more potent. Our data suggest that a clinical trial using RSK4 inhibiting floxacins in combination with chemotherapy may provide benefit to patients. So our next mission is to translate our findings into the clinic.